SIG. The chairperson for this session will be Dr. Chi Mok, and the speaker will be Prof. Sandra Navarra. Both of them will be online. Dr. Mok, hand over to you, please. Thank you.
So good evening, everybody. I'm Dr. C.C. Mok from Hong Kong. So I'd like to introduce our next speaker, Professor Sandra Navarra. Professor Navarra is the head of rheumatology at the University of Santo Thomas Hospital in Manila, Philippines. She is a steering committee member of Lupus Academy, executive board member of Asia Pacific Lupus Collaboration, and founding member of the Asian Lupus Nephritis Network. Professor Navarra is involved in a number of clinical trials of lupus and lupus nephritis and has published widely in the field of rheumatology, including systemic lupus, SPA, and other rheumatic diseases. She is also awarded the Most Distinguished Physician Award by the Philippine College of Medicine and the Master Award by the Aplar. So without delay, may I call upon Professor Sandra Navarra to deliver her talk entitled Recent Progress in SLE Treatments.
Thank you. Thank you, Professor Mok, for the gracious introduction and for this opportunity to share the exciting new developments in lupus. And good afternoon, good evening, greetings from the Philippines. So it would have been great if I can join you in person at Hotel Okura. And here are my disclosures. A better understanding of the molecular pathogenesis of lupus has paved the way for developing new and more effective therapies targeting B cells, type 1 interferons, and plasmacytoid dendritic cells, T cells, intracellular signaling, as well as toll-like receptors. The hyperactive B cells with production of autoantibodies are a hallmark of lupus, and B cell-directed therapies remain the anchor for drug development in lupus. Of these drugs, the chimeric CD20 chimeric monoclonal rituximab has a colorful history in lupus drug development. And although the Explorer, which is the non-renal study, and as well as the Lunar, which is the renal trial, failed to meet the endpoints, post-hoc analysis showed clinical and serologic benefits, particularly in special populations. And rituximab now continues to be widely used in clinical practice, although off-label, but especially for severe flares and refractory lupus. The majority of immunosuppressants used in lupus exert their therapeutic effects on B cells, plasmablasts, and long-lived plasma cells, but not on the vigorous antibody-producing long-lived plasma cells. Bortezomib is a proteasome inhibitor approved for treatment of multiple myeloma and has been used in a small study which has been shown effective for refractory lupus nephritis. And lately, zetomipzomib is a newer immunoproteasome inhibitor, and a phase 2 mission trial of zetomipzomib showed a clinical positive response in active lupus nephritis after 6 months. This is without high-dose induction therapy. Patients in the trial also experienced even reductions in the extrarenal manifestations of lupus. B-cell activating factor, BAF, or B lymphocyte stimulator, BLyS, is a key target molecule in B-cell pharmacotherapy for lupus. And belimumab, which is a fully human monoclonal antibody, selectively targets and inhibits BLyS, leading to increased B-cell apoptosis. And it is the first biologic approved for FDA for use in active SLE in 2011. Following the successful landmark belimumab phase 3 trials on BLyS 52 and BLyS 76, at least 2 other phase 3 trials have been performed, which reached the same SRI for primary endpoint at week 52. One assessed efficacy of subcutaneous administration, and the other assessed belimumab efficacy in a Northeast Asian lupus population, including Japan, China, and South Korea. Both trials yielded robust, significant outcomes, showing higher response rates with belimumab compared to placebo. BLISS-LN is a phase 3 RCT designed to specifically assess the effect of belimumab as add-on to standard of care in patients with active nephritis. And at the end of 2 years, more belimumab patients achieved the renal response efficacy of primary efficacy renal response, or PER, as well as the complete renal response, or CRR, endpoints versus placebo. And both endpoints were maintained to week 104 in the belimumab group. Moreover, the risk of kidney-related event or death at any time up to week 104 was actually 49% lower in the IV belimumab group compared to the placebo group. Now, findings from a recent pooled analysis of four phase 3 BLISS trials, which included 3,000 plus patients, and this is reported in Eurolupus T22 or last year, supports the use of belimumab and antimalarial agents in the prevention of renal flares. And the risk of flare was actually reduced in the IV belimumab 1 mg per kg group and IV belimumab 10 milligram group relative to placebo. But curiously, it's not seen in the subcutaneous belimumab 200 milligram group. The risk was also reduced with concomitant, as would be expected, antimalarial use. Atacicept is another biologic which works and blocks the effects of both BAFF, or BLISS, and its homologous B cell cytokine April. And the phase 2 April SLE Safety Committee, however, unfortunately, prematurely, and I would say incorrectly, terminated the higher atacicept dose. However, ITT showed this dose reduced the number of flares and time to first flare. Furthermore, the phase 2B ADDRESS trial showed long-term treatment with atacicept 150 milligrams in lupus patients had an acceptable safety profile with durable efficacy. Telitacicept consists of the extracellular domain of the human TACI receptor fused to human IgG, and the recombinant protein, which is similar to atacicept, was designed to inhibit both BLISS and the April. And in a phase 3 study of 335 patients with active lupus, and this was conducted in China, subcutaneous telitacicept once weekly significantly increased SRI-4 response at 52 weeks relative to placebo. When we summarize the immunosuppressive and biologic therapies for antibody-related mediated manifestations of lupus, it is important to note that effective curative options need to eliminate both plasmablasts and long-lived plasma cells. Future therapies, which may cover a broader scope of these cellular targets to include sustained B cell depletion by CD20 targeted chimeric antigen receptors, or what we call as CAR T cells, mesenchymal stem cell therapy, and the dual BLISS-APRIL inhibitor atacicept or telitacicept. In 2022, just last year, researchers reported outcomes among 5 patients. It's a small study who had severe and refractory lupus, and they were treated with anti-CD19 CAR T cell therapy. Within 3 months of treatment, 4 of these 5 patients actually achieved a SLEDAI-2K score of 0. The 5th patient had an improvement in SLEDAI-2K score from 16 down to 2. And all patients experienced a sustained drug-free remission during the median follow-up of 8 months, even after B cells reemerged. So although this was a small study, it is worth looking into, especially for patients who have refractory lupus. Type 1 interferons have been found to play a key role in the pathogenesis of SLE. And there are 2 main cells that are involved in the production of type 1 interferons. You have your plasmacytoid dendritic cell leading on now and stimulating your interferon responsive cells. And the therapeutics targeting various aspects of this interferon pathway are actually in different stages of development. Anifrolumab is a fully human monoclonal antibody that directly binds to the type 1 interferon receptor. Following a refinement in study design gained from a conduct of TULIP-1, the TULIP-2 phase 3 trial of anifrolumab successfully met trial endpoints and has been approved in the US for the treatment of adult patients with moderate to severe lupus who are receiving standard therapy. And this is now the only new treatment, the second new treatment approved for lupus in more than 10 years following the approval of belimumab. Plasmacytoid dendritic cells produce type 1 interferons, and blood dendritic cell antigen 2, or what we call as BDCA2, is a surface receptor that is exclusively expressed on PDCs and is a negative regulator of type 1 interferon. Yes, sorry about that. Let me share this. Yeah, and yeah, unfortunately, just experiencing. Okay, so let's go back. Yeah, apologies for the technical problems. Yeah, so we've seen a lot of exciting work being done. There's a lot of exciting action in the development of these new drugs. And as we go through these new drugs, including the newer drugs here, so talking about following anifrolumab, which is working on a direct inhibition of your interferon receptor, we now go into your the BDCA, which is found on the surface of your plasmacytoid dendritic cells. And here we have litifilimab or BIB59, which is actually a humanized monoclonal antibody that binds BDCA, resulting in downregulation
Prof.
of type
Sandra,
1
so sorry to
interferons.
disturb you at the moment. You have not shared your screen to us.
Oh, okay. Yeah. So sorry about that.
No worries. Thank you very much.
Okay. Yeah, sorry. So now you can
Thank
see my
you.
screen.
Yes, we
you.
can see. Thank you very much, Prof.
Thank you. Okay, so now I was talking about litifilimab, and litifilimab is actually B59. It's a humanized monoclonal antibody that binds BDCA2, resulting in downregulation of type 1 interferons. And here, last year, there were 2 New England publications which featured the litifilimab trials meeting trial endpoints. One was for cutaneous lupus erythematosus, and the other one was for systemic lupus erythematosus. Now, just to go into a bit into mechanism of action, it's that blocks BDCA2 on PDCs, and therefore that leads to the downregulation of type 1 interferon. And in this phase 2 trial involving lupus patients, litifilimab 450 milligrams administered sub-Q was associated with greater reductions in lupus disease activity, including active arthritis and skin disease. Immunoglobulin-like transcript 7, or ILT7, is a cell surface molecule which is specific to the plasmacytoid dendritic cells. And daxdilimab is a novel human monoclonal antibody that targets and binds to ILT7. Targeting ILT7 thus reduces the cells levels of your PDCs or plasmacytoid dendritic cells, therefore resulting now in decreased secretion of type 1 interferons. Phase 2 trials are now ongoing for daxdilimab in lupus, in SLE, and lupus nephritis, as well as other autoimmune diseases, including dermatomyositis. Lupus nephritis is always a hot item in lupus drug development on account of its high morbidity outcomes, even with standard treatment regimens. And illustrated here are the various therapeutic targets in lupus nephritis, including B as well as T-cell directed therapies and drugs which target the JAK-STAT signaling pathways. Voclosporin, or Lupkinase, is the newest generation calcineurin inhibitor, which is now FDA approved for lupus nephritis in 2021. Sirolimus, used in transplantation, blocks activation of T cells and B cells through mTOR inhibition and is being tried for extrarenal lupus. What about JAK-STAT inhibitors? Now, despite the promising results in the phase 2 trial, the phase 3 trial of JAK inhibitor baricitinib was unfortunately terminated due to its lack of efficacy. However, there were no safety issues. Orelabrutinib is an oral, highly selective, irreversible inhibitor of Bruton's tyrosine kinase approved in China for treatment of B-cell malignancies. In a phase 1b/2a dose-finding study in 60 patients with mild to moderate lupus, orelabrutinib added to standard background therapy and showed some promising clinical activity in lupus. Deucravacitinib is an inhibitor of a kinase called TYK2, which regulates signal transduction downstream of receptors for interleukin-23 as well as IL-12 pathways, and thereafter their type 1 interference. That is included now in the PACE-LE trial. This is a phase 2 study wherein up to 58% of patients treated with deucravacitinib versus 34% of placebo-treated patients met the primary study endpoint of an SLE responder index 4 after 38 weeks of treatment. Deucravacitinib also achieved meaningful improvements in all secondary endpoints, and safety and tolerability profile was generally similar to placebo. And we therefore look forward to phase 3 trials of this drug. And here's our good old antimalarials. So antimalarials like hydroxychloroquine have long been used and found very effective for lupus. However, the mechanism of action was not made clear to us until the recent decades. Now we know that these drugs primarily work by blocking toll-like receptor signaling. And moving on from there, we understand that toll-like receptors, especially toll-like receptors 7 and 8, play important roles in immune surveillance. However, activation of TLR7 and TLR8 by ssRNA released from tissue damage may contribute to the pathogenesis of lupus. TLR7 activation in particular enhances B-cell differentiation, increasing autoantibody production and immune complex formation. The TLR7 in PDCs stimulates type 1 interferon, as we heard earlier, and cytokine production, enhancing the immune response. TLR8 activation in PDCs, as well as TLR7 and 8 activation in monocytes and macrophages, stimulates production of pro-inflammatory cytokines. Notably, the increased intracellular NF-kappa B activation by TLR7 and TLR8 is actually associated and has been strongly associated with steroid resistance. And this, which is a major challenge in some lupus patients. So you might as well have a solution in these toll-like receptors. Afimetoran is a small oral selective inhibitor of toll-like receptors 7 and 8 that shows potent efficacy in preclinical models of advanced lupus. And the primary endpoint is SRI-4 at week 48. The estimated completion date of this phase 2 trial is by next year, May. And enpatoran is another investigational oral selective inhibitor of your toll-like receptors 7 and 8. And phase 2 WILLOW trial will now evaluate the glucocorticoid-sparing effects of enpatoran in cutaneous lupus and as well as systemic lupus erythematosus. It's actually the glucocorticoid-sparing activity will be assessed with mandatory dose tapering to a prednisone equivalent of less than 5 milligrams per day, which is a very, very relevant and pertinent endpoint, especially for our lupus patients, to bring down the steroids. The primary endpoint is the BICLA response at 24 weeks. At the risk of oversimplifying, I would like to summarize as follows. Targeting B cells actually include the first FDA-approved biologic belimumab, with now a 12-year track record, while rituximab continues to be widely used in clinical practice, as well as some now ongoing clinical trials. The human monoclonal antibody obinutuzumab is currently on phase 3 trials for lupus nephritis. Likewise, atacicept, telitacicept as inhibitors of both BLyS and APRIL, and the latest immunoproteasome inhibitor, zetomipzomib. Targeting type 1 interferons and plasmacytoid dendritic cells include drugs that have been developed like anifrolumab, which is only the second FDA-approved drug for lupus, and the recent litifilimab and daxdilimab, now on phase 2 to 3 trials. Primarily targeting T cells and to some degree in sync with B cells are the newest CNI voclosporin, approved also for lupus nephritis, in addition to the older CNIs like cyclosporin and tacrolimus, which are still widely used in clinical practice. mTOR inhibitor sirolimus, and the attractive novel CAR-T therapy. Intracellular signaling inhibition of JAK-STAT is done by baricitinib, which we know was promising in the phase 2 trial, but not in the phase 3 trial, but nonetheless, there were no safety issues. So we still look forward to that because interestingly, baricitinib has also been used in dermatologic cases like alopecia areata. And TYK2 inhibitor by deucravacitinib as in the PAISLEY trial, and Bruton's tyrosine kinase inhibitor, or BTK inhibition by orelabrutinib. And finally, toll-like receptor inhibition by the good old hydroxychloroquine, which is a must in patients with lupus, as well as the newer drugs right now on clinical trial, afimetoran and entaporan. Indeed, there are many more, including repurposed drugs, including secukinumab or combination drugs like rituximab with belimumab, or even novel drugs in the future. However, in lupus, we know that there is no one size which fits all. Indeed, despite the advances in drug development for lupus, there remain patients who simply refuse to respond to current therapies. And therefore, we need more of this. Lupus presents with an exceedingly wide range of manifestations, from quiescent but serologically active to major organ involvement. And no two lupus patients are ever the same. And it cannot be overemphasized that treatment should be highly individualized. We have struggled with lupus drug development for the last 30 years, but I am optimistic about the future. We will have new targets, we will have new strategies, we'll have more medicines, we'll have better prediction from biomarkers, and definitely better trial designs and endpoints. And this means better outcomes for our patients with better quality of life. Let's continue to find solutions for lupus. Thank you.
Oh, thank you very much, Sandra, for your excellent update on the treatment of lupus. Any questions online and also in Japan? I cannot see any online questions. Maybe I start by asking you a question. Coming back to the real world, we only have a belimumab, rituximab, and perhaps a newer CNI in US, but not yet available in Asia yet. So what will be your choice for patients who do not respond optimally to the standard of care? So any expert opinion on this belimumab, rituximab, and also the CNI?
we know that one of the challenges, especially in our part of the world, is not just this access to new drugs, but it's also the adherence to medications. So I would just first, number one, make sure that they're adherent, because we still have, and I'm sure you agree with me, CC, that we have effective therapies available with us. But the main roadblock that we have with our patients is still adherence. So we do with whatever we have available. And there are many repurposed drugs that are available, repurposed, including leflunomide for some cases, which we use in rheumatoid arthritis. And there have been studies that show that leflunomide can be effective for these patients as well. But I would like to stress it's so important to have them go in adherence for these patients to be adherent to their therapies.
I think it's important for ensuring treatment adherence and also early referral to a specialist for assessment. But practically, I was asked many times about the CD20 biosimilar and also the belimumab. You know, the biosimilar is now readily available in many Asian countries. What do you think? If a patient has refractory lupus manifestation, will you use belimumab or anti-CD20? Biosimilar, cut down the cost, yeah.
Yeah, well, whatever is available.
Mm-hmm.
We know that in the course of the development of the B cell, there are parts of the B cell development or maturation wherein belimumab works and rituximab doesn't work, and vice versa. That is why we have these combination therapies wherein you can use rituximab as an induction and then follow it up with maintenance with belimumab.
Okay, I see two questions online. So the first question is, will B-cell depletion therapy, with B-cell depletion therapy in lupus, did you follow the CD19 counts or the serum IgG level?
Yeah, there are studies that show that if you have patients who do not seem to have any depletion of their B cells are actually, they're not, these are the non-responders to your rituximab. I mean, it does make sense. So that may be one option of following them up with B cell depletion. If you find that your CD19 levels are not going down, then maybe your drug is not working, or maybe there are other mechanisms that you need to explore in your patient that are happening in your patient other than B cell mechanisms, pathomechanisms. So, and I see another. Do you recommend the step-by-step? There's a question. Do you recommend step-by-step treatment or multi-target treatment from start to avoid higher steroids or long-term effects? In general, I will answer. I'm sure you agree with me. The earlier you treat these patients, the better. And the more aggressive you treat these patients, the better. So I would go ahead and use multi-target. But just be careful with multi-target because you can go into safety problems, aside from the cost.
So, okay, how about the IgG level? So personally, I will check the IgG level as baseline before rituximab or belimumab therapy as a baseline. So if for repeated courses of rituximab, maybe some patient may develop hypogammaglobulinemia. So we may need to monitor for the IgG level. Do you agree with this?
Yes, because that is the one that's directly related to the risk for infections following B cell depletion therapy. So, instead of the CD19.
Yeah. CD19 is not available in many centers, but serum IgG is easily available. So I will check the baseline and monitor if the patient is on prolonged therapy with of the B cells like belimumab and CD20. So, any other questions online or at the meeting?
Can I ask a question?
Okay. Who is talking?
Prof. Ahmad?
Oh, Dr.
Yes.
Ahmad. Yeah.
Hi. Yeah, excellent talk. Wonderful. I have a question. What is your experience with belimumab in membranous nephritis?
I don't think there are any studies on membranous nephritis alone. So purely membranous nephritis. And even in the BLISS, even in the BLISS LN trials, I think they have excluded pure class 5 nephritis. So it has to be, you have to have proliferative lupus nephritis, which is class 3 and class 4. And they can be combined with class 5, but you have to have the class 3 and class 4, but not pure membranous nephritis. So there are no studies for pure membranous nephritis.
Thank you so much. An excellent session. Great, Sandra. Thank you.
So if there are no more questions, I will hand over to the next section, the chairman and also the speaker. So thank you very much for listening to this SLE talk.